Hello friends, this video on applications of biotechnology part 3 is brought to you by examfear.com. No more fear from exam. So now let us talk about some of the examples of genetically modified crops. So first here we will talk about BT. So what is BT? So BT is the name of it is basically derived from a bacteria. So there is a bacteria called Bacillus thuringiensis, and this bacteria produces BT toxin, and this is extremely important because we will see that how this BT toxin helps to produce genetically modified crops and how these genetically modified crops are insect resistant. So what does this BT toxin do? So let's see that. So this BT toxin has the capacity that it can kill insects. So that is how it acts as insecticide. Now the question is how this BT toxin, which is produced by a bacteria, will be incorporated inside a plant so that the plant can become resistant to insects. So that is what we will study here. So now this BT toxin gene in plants provide resistance against insects. Now this BT toxin is actually produced by bacteria. Now inside the bacteria, what is present? There is a cell which makes the bacteria. Inside that cell there are certain genes. So there is a particular gene which produces this BT toxin. So that BT toxin gene is extracted from bacteria, and the same BT toxin gene is inserted inside the plant. So if inside the plant you have the same BT toxin gene, what will happen? That gene is responsible for the production of BT toxin. So BT toxin will be produced in the plants as well. And what is the job of BT toxin? BT toxin acts as insecticide. So if this toxin is being produced in a plant, so the plant will be resistant to insects because it has an inbuilt insecticides in itself. Right? So the concept here is, the concept of genetic engineering here is that the BT toxin gene is extracted from bacteria and then it is inserted in the plants and then expressed in the plants. So basically with the help of genetic engineering, we can regulate gene expression. We can control where the genes need to be expressed. So in such type of plants which have the BT toxin gene, they do not need chemical insecticides out externally because they already have insecticides within themselves. And there are many plants where this type of genetically modified BT version has been produced. For one such example is the BT cotton. So this is the normal cotton and this is the BT cotton. So BT cotton is transgenic because it is genetically modified. So BT cotton is has inbuilt resistance against insects. Therefore, these will not be spoiled by insects. Whereas the normal cotton, they will get easily spoiled by the insects and they can easily get damaged. Also, this plant, in order to survive, would need external application of insecticides. So this is the advantage of BT cotton over normal cotton plant. Now, let us try to understand how the BT toxin actually works. So we will talk about the toxin in BT cotton plant. So BT toxin is toxic insecticidal protein. So it is definitely toxic. It is harmful, poisonous, insecticidal because it kills insects, and it, by structure wise, it is a protein. So how does it work? Now the one important question that might arise in your mind is, now when BT toxin is toxic in nature, when it is harmful in nature, then why doesn't it harm the bacteria when it is present inside the bacteria? Because the bacteria is producing BT toxin inside its body. So why is it not killing the bacteria itself? That is because the protein exists in its inactive form called protoxins inside the bacteria. So as long as it is present inside the bacteria, it is present in inactive form. So that time it is not poisonous. So this inactive form is called protoxin. But as soon as it is taken up by the insects, the protoxin gets converted into the toxin. That is, it actually becomes toxic and then it kills the insects. Now the question is, what actually triggers the activation of these BT toxin proteins? So once ingested by the insects, it turns into active toxins. Now what happens exactly? Let us try to understand that. Now, as I said, that Bt toxin is present inside the bacteria. So, how the bacteria produces this toxin? Which bacteria? It is Bt, that is Bacillus thuringiensis. So, this bacteria will produce certain protein crystals during a particular phase of their growth. I mean, this Bt toxin is not produced all the time. During one particular phase of their growth, the bacteria will produce these protein crystals. And these crystals will contain toxic insecticidal protein, which we call as Bt toxin. Now, they remain in inactive form inside bacteria called protoxins. Now, as soon as they are taken in, ingested means taken in by the insects or eaten up by the insects, they become active. And what activates them? The alkaline pH of the gut. So, what activates them? The alkaline, that is the basic nature. So, the alkaline pH of the insect gut. What is gut? Gut is nothing but the digestive tract. So, the alkaline medium of the digestive tract activates these proteins. So, these proteins need an alkaline medium to become activated. What happens once they become activated? As soon as it is taken up by the insect, they bind to specific receptors in the gut. When they are activated, they will bind to specific receptors. Receptors means those molecules which can receive or bind to other molecules. Now, as soon as it binds to these specific receptors, the insect will stop eating. Now, everything is happening in the gut. Gut is the digestive tract. So, digestive tract is closely linked to the eating process. So, the insect will stop eating. Now, when the insect stops eating, what happens over a period of time? So, there is no food coming inside. So, there is no energy being produced inside. And if the insect starves for a really long time, the insect will eventually die. So, the same thing happens. Over a period of time, the gut wall breaks down. So, that is the wall of the digestive tract breaks down. And as a result, the insect will die. Now, it is very common thing, right? If your digestive tract gets impacted so badly, in that case, the organism will die. So, this is how the Bt toxin becomes toxic. So, please understand this, because many people have this question in their mind that when bacteria is producing this Bt toxin, why is it not harmful to the bacteria? That's because it is present in an inactive form called protoxin. What is this protoxin? Like you would have heard about pro-insulin, pro-hormone, pro-enzyme. So, protoxin again is a chemical compound, and this compound becomes toxic only after it is altered in some way. That means altered in the sense only when its chemical structure is altered in some way. Only then it becomes toxic, and that toxic is toxin, Bt toxin. But when it is taken in by the insects, it will become activated. That is, it becomes toxic due to the alkaline pH of the insect gut. And as a result, it will break down the gut wall, the insect will stop eating, and finally the insect will die. So this is how Bt toxin works. Now, there are many crops which has been modified using the Bt toxin, using this Bt toxin genetic engineering concept. So, some of the examples we will share on the screen. Now, specific Bt toxin genes are specific to insect groups. So, different plants, there are different types of plants, or for example, cotton, corn, rice, potato, tomato. So all these plants have been genetically modified using the Bt toxin genes. So these genes have been transferred from the bacteria into these plants. But please note that it is not the same gene which can be transferred to any, which can be transferred to any plant and that can be resistant to any insect. It is not like that. Now, there are specific Bt toxin genes which are specific to particular insect groups. For example, and also please note that there is not just one Bt toxin gene which exists. There are several Bt toxin genes which are present. So mostly the Bt toxin gene is XII. So this gene is called XII, and this XII also, there are several types of XII. For example, there is XII1AC, there is XII2AB, there is XII-AB. So there are different XI genes present, and each of these genes, they are specific against a particular insect group. For example, this one, these two, XI-1AC and 2AB, these are good insecticides against bollworm. Similarly, XI-1B, this is a good insecticide against corn borer. So maybe this gene can be inserted into the corn plants because they are more easily attacked by the corn borer. So similarly, XI-1AC or XI-2AB genes can be inserted into those plants which are mostly attacked by the bollworm. So each gene has a targeted pest. So we have to decide which particular gene needs to be inserted into the particular plant depending upon the plant is easily attacked by which group of insect. Now, some of the plants which have been genetically modified using Bt toxin are cotton, corn, rice, potato, tomato. So these are some of the plants which are genetically modified using Bt toxin gene. So here you saw that how genetic engineering can actually help us to get or to produce modified crops. Modified in the sense crops which are more stable, which are more resistant to insects and pests so that they can survive for a longer period of time. There are no crop losses, the yield can increase, the nutritional value can also increase. But at the same time, you could see some of the disadvantages. For example, since they are insect resistant, that means that they have inbuilt insecticides. So that means they are also introducing insecticides into the food chain. So this was about the Bt toxin. Thank you. Please visit www.examfeo.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.